Hello and welcome to my channel today, Boo Snakes and More. Today we're going to be talking about what a species is. So please like this video and subscribe to the channel. A species is the basic unit of classification or a taxonomic rank of an organism, as well as a unit of biodiversity. It can be defined as the largest group of organisms in which any two individuals of the appropriate sexes and mating types can produce further offspring, typically by sexual reproduction. Other ways of defining species include their karyotype, DNA sequence, morphology, behavior, or ecological niche. In addition, pathologists use the concept of the chromospecies since follicular reproduction cannot be examined. The most recent rigorous estimate of the total number of species of electrolytes is between 8 and 8.7 million. About 14% of these have been described by 2011. All species except viruses are given a two-part name, a biomeme. The first part of a biomeme is the name of a genus to which the species belongs. The second part is called the specific name or the specific epithet in botanical nomenclature, also sometimes in zoological nomenclature. For example, boa constrictor is one of the species in the genus Boa, with constrictor being the specific name. While the definitions given above may seem adequate at first glance, when looked at more closely, they represent problematic species concepts. For example, the boundaries between closely related species become unclear with hybridization in a species complex of hundreds of similar microspecies, and in a ring species, also among organisms that reproduce only asexually. The concept of reproductive species breaks down, and each colonial lineage is potentially a microspecies, although none of these are entirely satisfactory definitions, and while the concept of species may not be a perfect model of life, it is still a useful tool to scientists and conservatists for studying life on Earth, regardless of the theoretical difficulties. If species were fixed and distant from one another, there would be no problem, but evolutionary processes cause species to change. This obliges taxonomists to decide, for example, when enough change has occurred to declare that a fossil lineage should be divided into multiple chronospecies, or when populations have diverged to have enough distinct character traits to be described as catalytic species. Species in higher taxa were seen from Australia until the 18th century as categories that could be arranged in a hierarchy. The Great Chain of Being. In the 19th century, biologists grasped that species could evolve given significant time. Charles Darwin's 1859 book on the origin of species explained how species could arise by natural selection. That understanding was greatly extended in the 20th century through genetics and population ecology. Genetic vulnerability arises from mutations and recombustion, while organisms are mobile, leading to geographical isolation and genetic drift. With varying selection pressures, genes can sometimes be in exchange between species by horizontal gene transfer. New species can arise rapidly through hybridization and polyploidy, and species may become extinct for a variety of reasons, with several mass extinction events occurring throughout history. Viruses are a special case, driven by a balance of mutation and selection, and can be treated as quasi-species. Definitions. Biologists and taxonomists have made many attempts to define species, beginning with morphology and moving towards genetics. Early taxonomists, such as Linnaeus, had no option but to describe what they saw. This was later formalized as the typological or morphological species concept. Ernest Mayner emphasized reproductive isolation, but this, like other species concepts, can be hard or even impossible to test for. Groups of organisms separated in space or time. Later biologists have tried to refine Mary's definition with the recognition and cohesion concepts, among others. Many of the concepts are quite similar or overlap, so they are not easy to count. The biologist R.L. Mandu recorded about 24 concepts, and the philosopher of science John Wilkins counted 26. Wilkins further grouped the species concepts into 7 basic kinds of concepts. 1. Amogrammar species for asexual organisms. 2. Biospecies for reproductively isolated sexual organisms. 3. Ecospecies based on ecological niches. 4. Evolutionary species based on lineage. 5. Genetic species based on gene pool. 6. Morphospecies based on form or phototype. And 7. Taxonomic species, a species as determined by a taxonomist. Typological or morphological species. A typological species is a group of organisms in which individuals conform certain fixed properties. A type which may be defined by a chosen normal species, so that even pre-literate people often recognize the same taxon as do modern taxonomists. Modern-day field guides and identification websites, such as Naturalist, use this concept. The clusters of variations or phototypes within specimens, such as longer or shorter tails, would delineate the species. This method was used as a classical method of determining species, such as with Linnaeus early in evolutionary theory. However, different phototypes are not necessarily different species, e.g. a 4-winged diaphason born to a 2-winged mother is not a different species. Species named in this manner are called morphospecies. In the 1970s, Robert R. Sloga, Theodore J. Culver, and Peter Snicks proposed a variation on the morphological species concept. A prothotic species defined as a set of organisms with a similar phenotype to each other, but a different phenotype from other sets of organisms. It differs from the morphological species concept in including a numerical measure of distance or similarity to cluster entries based on multivariate comparisons of a reasonably large number of prototypical traits. Recognition and cohesion species. A mate recognition species is a group of sexually reproducing organisms that recognize one another as potential mates. Expanding on this to allow for post-mating isolation, a cohesion species is the most inclusive population of individuals having the potential for phototypical cohesion, though intercyclic cohesion mechanisms. No matter whether populations can hybridize successfully, they are still distinct cohesion species if the amount of hybridization is insignificant to completely mix their respective gene pools. A further development of the recognition concept is provided by the biomestic concept of species, genetic similarity in barcode species, single locus barcoding. In microbiology, genes can move freely even between distantly related bacteria, possibly extending to a whole bacteria domain. As a rule of thumb, microbiologists had assumed that members of bacteria or archaea with 16S rubinism RNA gene sequences more similar than 97% of each other need to be checked by DNA hybridization to decide if they belong to the same species. This concept was narrowed in 2006 to a similarity of 98.7%. The 16S sequence is an example of a single locus, which is simple enough for non-specialists to apply and in most cases significant to distinguish species. Using a single easy-to-use locus to distinguish taxonomy is called DNA barcoding. One of the barcodes for eukaryotes is a region of mitochondrial DNA within the gene for chromosome C oxidase. A database barcode of life data system contains DNA barcode sequences from over 190,000 species. However, scientists such as Rob D. Seller have expressed concern that classical taxonomy and DNA barcoding, which they consider a misnomer, need to be reconciled as they delimit species differently. Genetic integration mandated by endomycosis and other vectors can further make barcodes inefficient in the identification of species. A single locus can be a good proxy of time of development, assuming the chosen locus evolved like most of the rest of the genome. This assumption can be broken by horizontal gene transfer affecting the locus itself, rapid modes of evolution separating biological species. Speculation over a short time span would also decouple the species concept from time itself. Among bacteria, there are several cases where very different genomes share 99.9% 16S identity. Mitochondrial consumption using multiple, usually fewer than 10, LOCI for comparison provides more polygenetic signal compared to comparing versions of the same loci of the same LOCI, as more mutations can be captured. As a result, it provides improved taxonomic resolution compared to single locus comparison, giving results more similar to the expensive gold standard of whole genome comparison at a small increase in cost. Even when whole genomes are available, there are good reasons to only compare select genes. Many genes are not universally found in all genomes, so they've applied limited taxonomy signal while still adding to the computational cost of comparison. In situations like this, tens of hundreds of loci may be extracted from each genome and used together. As with comparisons with fewer LOCI marker genes, used for this purpose should be genes with low rates of horizontal transfer and gene duplication, few known instances of horizontal transfer, and high occurrence in the sampled genomes. With polykinetics, marker genes can be used to delimit taxonomy down to the genus level, with whole genome comparison reserved to separate species from each other. Whole gene comparison. The surefire way to capture all gene flow among populations is to compare their entire genomes. The average nucleotide identifier INA method qualifies genetic distance between entire genomes using regions of about 10,000 base pairs, With enough data from genomes of one genus, algorithms can be used to categorize species. As for Palidamas aviarum in 2013, and for all sequenced bacteria and Archaea since 2020, observed in ANI values among Percredo sequences appear to have an AIN gap at 85 to 95%, suggesting that the genetic boundary suitable for defining a species concept is present. A polygenetic or catalytic species is the smallest agitation of population, sexual, or lineages asexual, distinguishable by a unique combination of character states and comparable identities. The equimaker basis observed character states provides the evidence to support hypotheses about evolutionary divergent lineages that have maintained their hereditary integrity through time and space. Molecular markers may be used to determine diagnostic genetic differences in the nuclear or mitochondrial DNA of various species. For example, in a study done on fungi, studying the neuroquadrature characteristics using catalytic species produced the most adequate results in recognizing the numerous fungi species of all the concepts studied. Versions of the polygenetic species concept that emphasizes monolithicity or dinosaurity may lead to splitting of existing species, by example, in Barra Verde, by recognizing old subspecies as species despite the fact that there are no reproductive barriers and populations may integrate morphologically. Others have called this approach taxonomic inflation, diluting the species concept and making taxonomy unstable. Yet others defend this approach, considering taxonomic inflation projective and laboring in the opposing view as taxonomic conservationism, claiming that it politically expanded to split species and recognize smaller populations at the species level, because this means they can more easily be included as endangered in the IUCN Red List and can attract conservation legislation and funding. Unlike the biological species concept, a catalytic species does not rely on reproductive isolation. Its curricula are independent of processes that integrate in other concepts, therefore it applies to asexual lineages. However, it does not always provide clear-cut and involuntary satisfying boundaries between taxonomy and may require multiple sources of evidence, such as more than one polymorphic locus, to give plausible results. Evolutionary species. An evolutionary species suggested by George Gaylord Simpson in 1951 is an entity composed of organisms which maintains its identity from other such entities through time and over space, and which has its own independent evolutionary fate and historical tendencies. This differs from the biological species concept in embedding persistence over time. Lilly and Mandine stated that they see the evolutionary species concept as identical to Will Hanning's species as lineages concept, and asserted that the biological species concept, the several versions of the phylogenetic species concept, and the idea that species are the same kind as higher taxonomy are not suitable for biodiversity studies. With the intention of estimating the number of species accurately, they further suggested that the concept works for both asexual and sexual reproducing species. A version of the concept is Kevin D. Quizzo's general lineage concept of species. Ecological species. An ecological species is a set of organisms adapted to a particular set of resources, called a niche, In an environment, according to this concept, populations from a discrete pathogenic clusters that we recognize as species because of ecological and evolutionary processes controlling how resources are divided up tend to produce these clusters. Genetic species. A genetic species is defined by Robert Baker and Robert Bradley as a set of genetically isolated interbreeding populations. This is similar to Mary's biological species concept, but stresses genetic rather than reproductive isolation. In the 21st century, a genetic species could be established by comparing DNA sequences. Earlier, other methods were available, such as comparing ketotypes, sets of chromosomes, and allelose enzyme variants. Evolutionary significant unit. An evolutionary significant unit, ESU, or wildlife species is a population of organisms considered distinct for purposes of conservation and polytriacal with only comparative anatomy, morphologically and heredity from fossils as evidence. The concept of chronospecies can be applied during analogies to evolution, not necessarily involving branching. Some polykinetics seek to identify a sequence of species, each one derived from a polyraptor extinct one before, through continuous, slow, and more or less uniform change. In such a time sequence, some pathologists assess how much change is required for a morphological distant form to be considered a different species from its ancestors. Viral querospecies. Viruses have enormous populations, undoubtedly living since they consist of little more than a string of DNA or RNA in a protein coat and mutate rapidly. All of these factors make conventional species concepts largely unacceptable. A viral quasi-species is a group of genotypes related by similar mutations competing with highly mesosonic environment and hence governed by a mutation selection balance. It is predicted that a viral quasi-species at a low but evolutionary natural or highly connected that is flat region in the fitness landscape will outcompete a quasi-species located at a higher but narrower fitness peak in which the surrounding mutants are unfit. The quasi-species effect, or the survivor of the flattest. There is no suggestion that a viral quasi-species resembles a traditional biological species. The International Committee on Taxonomy of Viruses has since 1962 developed a universal taxonomic scheme for viruses that has stabilized the viral technology. Mayer's biological species concept. Most modern textbooks make use of Ernest Mayer's 1942 definition known as the biological species concept as a basis for further discussion on the definition of species. It is also called a reproductive or isolation concept that defines a species as groups of actively or potentially interbreeding native populations which are reproductively isolated from other such groups. It has been argued that this definition is a natural consequence of the effect of sexual reproduction on the dynamics of natural selection. Mayer's use of the adjacent potentially has been a point of debate. Some interpretations exclude unusual or artificial matings that occur only in captivity, or that involve animals capable of mating, but do not normally do so in the wild. The renewing species concept extends Mayer's biological species definition to also include amongst asexual organisms. It extends the concept of interbreeding the glue binding separate individuals into a species to the concept of renewal cohension, the glue that works both for sexual and amigous organisms. Renewal cohension is the resurrection of genetic diversity of a group of related individuals as a result of their recent origin from a common ancestor. This limitation on genetic diversity results from the efforts of genetic draft explained by correlation theory, which is significant to resurrect genetic diversity even in the absence of selection. Reproductive isolation is anti-interbreeding, and a prerequisite for speculation is extended to renewal isolation, inclusive of both sexual and amongst organisms. A renewing species is the most incohesive group of individuals which are united by renewal cohension, i.e., one are reproduced in a specific ecological niche, and two are genetically similar due to recent origin of their genomes, as whole uninterrupted DNA stretches, or in parts, from genomes of ancestral individuals. Applied to sexuals, the concept of renewal species is reduced to Mayer's biological concept. Apart from the connection with Mayer's biological species concept, renewing species borrows its mechanism of cohension from Temperton's cohension via genetic draft, one of the cohension species mechanisms described. The species problem. It is difficult to define a species in a way that applies to all organisms. This debate about species concepts is called the species problem. The problem was recognized even in 1859 when Darwin wrote in On the Origin of Species. I was much struck how entirely vague and ambiguity is the distinction between species and variation and varieties. He went on to write, no one definition has satisfied all naturalists, yet every naturalist knows vaguely what he means when he speaks of a species. Generally, the term includes the unknown element of a distant act of creation. Problems with simple definitions of species. Many authors have argued that a simple textbook definition following Mayer's concept works well for most multicellular organisms, but breaks down in several situations. When organisms reproduce asexually, as in single-celled organisms such as bacteria and other prokaryotes, and pathogenetic or apometic multicellular organisms, DNA barcoding and phylogenetics are commonly used in these cases. The term quasi-species is sometimes used for rapidly maturing entities like viruses, when scientists do not know whether two morphological similar groups of organisms are capable of interbreeding. This is the case when hybridization permits substantial gene flow between species. The ring species were members of adjacent populations in a widely continuous distribution range, interbreed successfully, but members of more distant populations do not. Species identification is made difficult by discordance between molecular and morphological investigations. These can be categorized as two types. One, morphological multiple lineages, e.g. morphology, convergence, cryptic species, or one lineage and one lineage multiple morpholineages. Phototypic plasticity, multiple life stages. In addition, horizontal gene transfer makes it difficult to define a species. All species definitions assume that an organism acquires its genes from one or two parents, very like the daughter organism, but that is not what happens in HGT. There is strong evidence of HGT between very dissimilar groups of prokaryotes, and at least occasionally between dissimilar groups of eukaryotes, including some crustaceans and Echinomedia.